lecture. And so today we're going to be looking at lecture 9. It will be focusing on microbial diversity. And it's really a survey of all, well, a brief survey of all the microbes that living around us or in extreme environment. Okay, so we have the microbes generally can be divided into bacteria and archaea. And then you have eukaryote microbes as well, single cell, right? And which we discuss in the lecture 1. So within bacteria, they can be further separated into 17 phyla. Okay, so now you're going to learn a little bit of terminology. Phyla is the plural form, just like a bacteria versus bacterium, right? And bacteria is a plural form, multiple. And so phylum 1, phylum 2, all the way to phylum 17. And within archaea, which is the microbe, the domains, remember the 3 domains, the eukaryote, bacteria, and archaea. And so archaea can be further divided into 3 phyla, and which we'll talk briefly about them. In general, we know more about bacteria, we know less about archaea. It's because archaea tend to live in extreme environment, and bacteria tend to live in less extreme environment. Now, the problem with studying bacteria living in extreme environment is that it's hard to create a condition to get them grow. One of my first research project after I graduated from college was to study a bacteria, go out and isolate a bacteria from hot spring. The goal there was to isolate the DNA polymerase for PCR. So the bacteria, the hot spring is 68 degrees. So we looked around trying to find a growth condition that a shaker incubator that can go to 68 degrees and maintain. And the only one we can find was a water bath that can get to high temperature. The problem is the water bath with a shaker, it cannot be sealed. And so the water, keep water at 68 degrees Celsius, the water evaporates really fast. And very soon the room feels like a sauna, right? So it was difficult. The point is that it's more difficult to study, and so now we ended up we know a little bit less about the archaea. Okay, so if we were to look at the bacteria, the 17 phyla, and these are the 17 phylas. Okay, so starting the biggest is called the proteobacteria. That's phylum 1. And then you have phylum 2, and so on and so forth. So what I would do is that in today's lecture, I'll spend a lot of time talking about proteobacteria. And these are the bacteria lives really in our environment, I mean, share the same environment. So we encounter them a lot, and we have them on our skin, in our gut, and in our eye, and it's all over. And then there's gram-positive bacteria as a group. So those 2 are the major groups I'll be talking about, but I'll cover a little bit about the ones that I use, I marked it as a star. So it's important that you understand the bacteria can be further grouped into these 17 groups, okay? And so now we're going to take a close look at the proteobacteria. And the proteobacteria can be further grouped into these subgroups: the beta, alpha, zeta, epsilon, and delta. Now, these are separated based on the 16th rRNA gene. So this is the most common way of separating bacteria. So I'm going to pause a little bit to talk about what's 16th rRNA. So if you remember, in the last last section, we talked about translation, right? Translation. Sorry. So translation is carried out by ribosome, and we mentioned at some point the ribosome has ribosome RNA. And bacteria has 3 ribosome RNA. The small subunit of ribosome has the 16th rRNA. And the large unit has the 23 and the 5S. So it has 2. And the 16th rRNA has a loop that binds to the upstream of the open reading frame. That's the ribosome binding site. And the ribosome binding site, it takes place is because it's needed because this rRNA is actually being used to bind the sequence to form the complementary sequence. And so that the 30S ribosome RNA can bind to the correct or the start of the open reading frame of the gene to start the translation, right? Now, so those are the things that we discussed previously, but we didn't really had the time to explain is the rest of the 16th rRNA is actually being looped around inside the ribosome. Okay, the ribosome is a protein complex, right? So there's lots of proteins, small proteins, and the small protein fits together to form a complex. And this formation of a complex is also helped by the ribosome RNA. So the way you think about this is the 16th ribosome RNA is a string that ties all these proteins together. Now, if you think about a string, the function of the string is to tie these things together, right? Just like your shoestring, right? Your shoestring is, the function is tie your shoes together. The color doesn't really matter, right? And so if we were to identify people by shoestring, what would you do? We can make kids go home and we'll make each of you to color your shoestring. And I'm sure you will come up with a very, very creative color scheme so that your pattern of your shoestring will be different, so that each of you can be coded. We can recognize, we can link one particular shoestring pattern to one of you, right? So you'll be recognized by the patterns of shoestring, right? And so we have 23, or we have 23 students in the class. For sure, we can recognize each of you individually, right? So that's why the shoestring or the 16th sRNA can be used to identify individual bacteria because they are unique. They are unique. However, the microbes in the world are slightly different from your students in our class, and they are related, almost like your family. You have siblings, right? And you have parents and you have grandparents. And so if we look at the genetics trees, the genes that are among you, and that we can determine based on um, how similar your DNA is, right? Obviously, your siblings and are very close, and they all derive from your parent. So the way you think about this is that, okay, so this is a sibling one, sibling two, and that's parent, your parent, right? And then that's another your that's your parents' brother. And then if you go to your grandparents, and so soon maybe this will be the second cousin. This will be first cousin. Going over here, maybe second cousin, right? And so this is a way of sorting out the how related the bacteria is. Now, why is that important? Now, many of you are interested in going to medical field, either medical school or PA school, right? And so disease, so the genetic background for people are important, for patients are important, right? So now we're going to the family history. Some disease are genetically related. That's because the gene has influence. And so this is also the reason that the bacteriologists use genetics to group bacteria together, because they will have a lot more things in common. Okay. And so a higher percentage of genes are in common. So for example, if you look at E. coli and Vibrio, that's the one that causes cholera, right? And the disease. And so then you can think about, okay, these two microbes have a lot of genes in common, and so we can study them together. And so that's the red, and so that is the gene tree. Okay, so so microbes can be further grouped based on their genetics and specifically the 16th rRNA genes, because they all have rRNA, ribosome RNA. They can use, and there are other genes, there are other proteins that can also be used to group them, and so so that's the genetic tree, and that's one way of separating. Yes, what's the question? Go ahead.
For gamma, the bacteria at the most top is the oldest one?
No, it's not the old. So this tree is not the oldest. The oldest is here. This goes by the genetics, the distance. And so it measures the distance. So the top, it's going follow up the tree. So the first one and the second one are the close relatives, because you see that they diverge here. And then you go up, and so these three are closely related. So gamma as a group are closely related. Then you have beta. They all share the same ancestry, right? Because beta is easier to see. The beta is more close. See, does that make sense?
Yes.
Okay, thank you. So now I'm going to summarize what we just talked about. Is that the proteobacteria can be further grouped because proteobacteria has hundreds of them. Proteobacteria can be further grouped using genetics or using phylogeny into at least 5 different groups, okay, at the moment. And each group has a representative. So, because you were thinking about group, it's just harder to think about a group without a representative, right? So for me, so you can go into each of the group to pick out one bacteria that you like. But for me, this is the group. These are the microbes I like. The alpha proteobacteria is rhizobium. I use rhizobium to represent alpha bacteria. It's because this is the bacteria I've been studying in the last 23 years. And then I like the bacteria called Burkholderia that I studied for 5 years as a graduate student. I like that one. And then I use gamma, E. coli to represent gamma, and so on and so forth, right? So this is my list to represent each of the 5 different groups. You can come up with your own after you go through each of the bacteria. So that's one way of grouping, separating bacteria into subgroups, right? But if you think about our class of 23, right? We can group our class into based on based on what grade or what level you are. Are you junior? Are you senior? Are you graduating senior? Right? We can group you based on how soon you'll graduate, right? And then the other is we can also group you based on, well, have you had biochemistry? Say yes, no, yes, no, right? So a group can be further any small group of people can be further separated into different categories, right, for different purpose. So if I would say, okay, I want to know how do you who will graduate immediately, so I can focus on tell you about graduation requirement. Then I need to know, okay, I'll need to separate the class into how soon you're going to graduate, and then I'll pick the ones that you're going to graduate, and then I'll talk to you, right? But then there are some other occasions I need to talk to you about things like biochemistry. And so I wanted to know if you had biochemistry. If you hadn't, so for the group that had biochemistry, then you expected to know amino acids and all those. But for the students who haven't taken biochemistry, they may less familiar with amino acids and all that. Then I need to talk to this group. Okay, so based on the purpose, and anything can be further separated. So for proteobacteria, historically, it was always separated based on metabolic characteristics, because until 1980s, or until about 10 years ago, the phylogeny is difficult to get. Sequencing DNA is very, very difficult. Okay, how difficult is to sequence DNA? If you heard about Sanger sequence, manual sequence, okay, I spent 2 years, actually 3 years, sequenced a 2 kb DNA piece, a gene, but now like a whole genome, a million base pair can be sequenced in a day. Okay, that's just the advance. So from before 1980s, how do bacteria get further grouped? well, based on the metabolic characteristics. And so these are they're further grouped into 17 metabolic groups. Okay, so before I go into this, you understand that proteobacteria is a collection of bacteria. Okay, and the reason proteo is multiple shape, and when people first look at the bacteria and saw, oh, bacteria have different sizes and shapes, and so they call them proteobacteria. But then they realize, oh, individual bacteria species, actually, if you purify to single species, individual bacteria maintains the size and shape. And so that's why I say E. coli is, the size of E. coli is, come on, you forgot? What's the size of E. coli? Yeah, okay, great, great. Thank you. 1/3, always have, right, right, thank you. 1/3 micrometers. Okay, you should be reciting this in your sleep. If um, that will be a good success, count towards my success, right? Um, okay, so the next period of time, I wanted to talk about the individual characteristics, metabolic groups. And because the reason you would do that, we still talk about these metabolic groups, is that they based on how uh, they metabolize, how do they do things, right? Because partially because we want to grow them, we want to know how they will grow them, how we can stop them from growing. And so these groupings are more practical, so we're still using them. Okay, it's almost like we want to group the students, our class, into how fast you run. Are we fast runner, medium runner, right? And why we need a fast runner? Well, because imagine if I'm in my in your teaching lab and I need something from Davis Hall, and I'm going to send someone from Science Hall to Davis Hall, and I want it to be fast, right? I'm just joking. So they so we're going to first look at the first group that's most important to us is the group with E. coli. So E. coli belongs to uh, enteric bacteria, and the common feature is that it's gram-negative and non-spoilating. This is E. coli, rod-shaped, facultative, aerobe, meaning it can do aerobic respiration and fermentation, and it has simple nutrient requirement. And this is actually pretty important. Simple nutrient environment. That means it can grow in almost any conditions, right? That also means it can produce everything it needs. And so if you think about us, do we have simple nutrient requirement?
No.
Ours is complicated. Why is ours is complicated? What things we cannot make? Anyone remember?
We can't make our own food. We're not autotrophs.
Oh, vitamin C is, yeah, vitamin B and B12, lots of vitamins. Um, and what about amino acids? Can we make all the amino acids? Right. We rely on food for 8 different amino acids, right? Um, I only remember the number, but I don't remember which one. So this is simple. For bacteria, though, they don't eat food. They live in the environment. They have to make everything. So that's what the simple requirement. But this is also important because if you just have water and glucose, and that gets spoiled because some microbes will be able to use glucose and some residual nitrogen, and be able to just grow, okay, and then spoil whatever you have. And then you have from E. coli has the fermenting sugars, and well, not in every environment, but the simpler the requirement, the more competitive they are. They can grow in more environment because not all environment have all the nutrients. So um, the microbes have simpler requirement can outcompete the other ones. So fermenting sugars to many different end products, and that's important feature because we'll come back to that. And we know the one that fermenting sugar to a single product is yeast. Okay, yeast compared to E. coli, yeast just ferments sugar to ethanol. That's it. And that's why you want to use yeast to make wine. You don't want to use, and when you make wine, you don't want your E. coli there. I'll get to that in a second. Um, it's with or without Petrotrichosis flagella. That's having bacteria all over the place. The best known representative is E. coli. Okay, and that's a family name. And then the other is Salmonella. Have you heard about Salmonella? When was the last time you heard about Salmonella? Uncooked chicken, yes. Well, it's causing problem typically in like meat patio recall or salad recall or something, right? Because they are they're similar. So without knowing what salmonella is, how would you be able to guess what how would you want to grow salmonella? The temperature, the nutrient requirement, and what possibly they might be grow. And so that's the advantage of grouping them based on metabolic features. So without knowing much about salmonella, if I say, well, okay, I'm gonna grow salmonella, and the first thing I do is, well, I know how to grow E. coli. I'm just grow E. coli, grow salmonella the same way as I grow E. coli because they're in the same group, right? That's the one of the reason you want to have metabolic grouping. So, Petrotrichosis flagella is the one that the flagella could be located in different parts. Remember, uh, let's see if I can draw. This is terrible. I need to get a pad. Well, Petrotrichosis is like this, right? Have flagella all over the body. Remember? That's the one. Okay, that's that's what the flagella look like. So this is this is to tell you about fermentation by E. coli, and compared to yeast, which produce ethanol, and E. coli can ferment and take glucose and make take glucose and make all these end product, acetate, succinate, and lactate, and some other product. And the end result is, well, ethanol tastes okay, right? That's in your beer, in your wine, but these acetate is really bad taste. And so that's why you don't want these microbes in E. coli in your wine. And then you have in the lab, we should have you had the demonstration experiment that shows you the fermentation? Have you made the observation? Yes, thank you. So you've seen this inverted tube because they're not clear on these in this picture. The acid changing pH, and that's obvious. Okay. So back to E. coli, and if we, it's everywhere, and it's inhabits of intestinal tract of humans and all kinds of warm-blooded animals. And one of the many things it does is it produces vitamin for hosts. And as a microbiologist, and I'm always curious, I'm thinking about, okay, so we allow these E. coli to grow, to colonize our intestinal tract. It probably is providing something for us, right? We know for sure if E. coli isn't there, if we take you heard about C. diffs, right? People with C. diff, Clostridium. It's a Clostridium bacteria. If you take too much antibiotic and kill all the microbes in your gut, and then your intestinal tract is colonized by Clostridium, you actually have a problem. Okay, so the natural E. coli, in addition to produce vitamin, it protects it prevents other pathogens from colonizing your intestinal tract, and to provide what we call the native protection. And so that's the main function. And I'm sure the more we study, we'll learn more about what do they do. We actually do talk about that in my research lab. One of the challenge that we face is, we don't know what they do in your gut. So the question is, E. coli is a good bacteria? Yes, in general, it's a good bacteria. Hold 1 second. So the one of the challenges, we don't actually know what's the growth condition in the intestinal tract. So we know it's anaerobic, right? But we don't know what about pH? What about liquid? How fluids are working in the intestinal tract, right? And so there are lots of things that limiting, and it's not like you can just poke a hole in your gut to just test, right? So it's difficult. And you can say, well, we can do colonoscopy, but colonoscopy really just cleans out the bacteria, cleans out your content before you can go in. So it's not the real environment that you're looking at. Okay, so the point is, E. coli, we have a lot to learn about what they do in natural environment, right? Nowadays we're thinking about the colon cancer, and there are also Crohn's disease. Crohn's disease. I don't know if you heard about it, which we'll talk about. So there's lots of unknown, but the main thing, the E. coli is the major intestinal tract inhabitor. Now, there are some E. coli are pathogenic. Okay, so let's see. So have you heard about an E. coli that is pathogenic? Bloody diarrhea? No? Have you heard about an E. coli that can cause bloody diarrhea? 0157H7. Have you heard about this E. coli? Okay. Okay, so you guys wanted to go to medical school, and there were lots of, or PA school. There's lots of microbes, and I'm hoping after this class, your ear will be, or your eye will be much more sensitive in whenever you hear about bacteria causing problem. 0157 H7, I think I typed in 0, should be O. O. Okay, so E. coli, this E. coli can cause bloody diarrhea, and it has a different O antigen. Remember, the gram-negative bacteria has these lipopolysaccharide at the very end that is unique. And so that's how the E. coli is being differentiated using serotype. Before we have any other method of differentiating, we use serotype that has antibodies that recognize different bacteria, different E. coli. And this is one of the ways we differentiate. They have different polysaccharide end. Now, there was an earlier question about E. coli. Well, good E. coli, yes, that's the Shigella toxin producing bacteria. Now, it's the point that perfectly prompt is that E. coli normally is a good guy. Well, at least a harmless bacteria. However, if the harmless bacteria pick up a dangerous weapon, and then they become pathogenic, right? So this E. coli, for example, we just had they raised the question about Shigella toxin. They got a toxin gene from another bacteria, the gene, and now this E. coli stopped producing toxin, a different bacteria's toxin. So now this E. coli all of a sudden becomes pathogenic. Why? Well, because they arm themselves with a knife. So now become a bad guy. It's okay. So so in the advanced class, if you read a little bit more, you would find that E. coli actually acquired this 0157. Now we know it acquired several toxin genes from different bacteria, and and so then it becomes pathogenic. Okay, so now there was a good question about why do you do that? Why would E. coli get a toxin gene? Is there advantage? Well, yes, they get a gene by conjugation or even transduction or lysogeny, meaning the phage lysogenized into the chromosome. Those all happen. But the question to all of you is, why? Why would E. coli get a foreign toxin gene? So the way you think about this is there must be some growth advantage, right? Because remember, the only thing that the bacteria wants to do is, I'm going to take a sip of water, let you think about. Reproduction, excellent, excellent. Thank you, thank you. I'm very happy. Getting some message through. So the only thing E. coli or any microbes care is reproduction. I mean, that can apply to higher levels of animals too, right? Animals, what do you think animal care? They only care about reproduction, right? So if you think about E. coli, now what would they need for reproduction? They need energy. So is there advantage for E. coli to cause bloody diarrhea? What happens to bloody diarrhea? Well, meaning they somehow injured your intestinal lining, so you start bleeding, right? What's in your blood? The blood has all the nutrients. Yes, glucose, obviously. The glucose and all the nutrients, right? Isn't that a best? Is that better than whatever you eat? Whatever left in the intestinal tract. Think about what E. coli is facing in the large intestinal tract, right? So the intestinal have the small intestinal, large intestinal. Small intestinal, you guys know, is all lactobacillus, lactobacilli, and enterococcus, right? Because it's acidic. Only the large intestinal tract is neutral, so that's where E. coli live. So all the main nutrients, the sugars that you eat, they're all being absorbed by small intestinal, and what's left is pretty minimal nutrient, right? So now if you can cut open the causing a reaction in your intestinal tract lining, and your blood start leaking into the intestinal tract, is that better for the E. coli? Right? It's obvious it's better for E. coli. Well, it's horrible for us. And so those E. coli then will have growth advantage, right? And so they will take over and then grow very, very fast, and they reproduce. So you think about bacteria, any bacteria, if they get a foreign gene or foreign toxin that provides a growth advantage, and those bacteria will populate, those population will increase, right? Eventually they will take over. And that's how evolution actually eventually happens, right? The genes gets more and more growth advantage. Okay, so that I'm using this as a discussion to bring you to explain the concept of what's happening in the nature and the advantage. Okay, so that's about E. coli. And let's continue the survey, wandering around the microbial world. And this is some bacteria that can have lots. Oh, this is the bacteria I want to show you about, flagella. Petrotrichoides has flagella all over the body. And the flagella tend to fall off. So you will see some flagella floating. And when bacteria have flagella, they tend to form these ring, like when they grow on the surface, they will spread out and then for these interesting pattern. The next major group of metabolic group that I wanted to tell you about is called the Pseudomonas. The Pseudomonas is a straight and chemo-organotrophic. And so it gets energy from organics, okay? And it's an aerobic rod with polar flagella, and that's unique. It's definitive. It's aerobic rod with polar flagella. And what's interesting about this bacteria, and in terms of metabolism, is it's nutritionally very diverse. It can eat almost anything. It can convert many different things into energy. Like the way you think about this is, well, you obviously this bacteria can use glucose as energy source, but if you give this bacteria tetracycline, do we know tetracycline? It's antibiotics, right? And if you give tetracycline to this bacteria, well, this bacteria actually use that as a carbon source. You say, voila, food. And on the other hand, you're thinking tetracycline is killing them. The bacteria says, I'm not going to be killed by tetracycline. These are great food for me. All right. So that's one example. And so you could find them because of its nutritionally so diverse, and you can find them in water and soil and everywhere. And it's an opportunist pathogen for human and plant. When we say opportunist pathogen, is that it doesn't actually have the like a regular pathogen. You have it, they will cause disease. They need some help for infection. So, for example, if you have this bacteria on your hand, it's okay. Even if you dip your hand into a bacteria solution, it's okay. Well, because as long as your hand doesn't have a cut, that's okay, because they cannot break through the skin. Okay, and so so that's the opportunist pathogen. Similarly for plant as well, and if the plant has a cut, then it will go in, it will infect. Now, this is an opportunist pathogen for the CF patient. Okay, so people have a long disease, and that they can cause they can colonize the lung of those patients, and then in addition to this bacteria, and there's another bacteria called Horderia cepacia. This infects onion. So if the onion rots, and that's where this bacteria is. If you get onion and it's rotted, and the rot has a lot of these bacteria. And one of the reason I highlight this is because I studied 5 years on this bacteria. So I just Googled it, and if you want to try, if you type in Cepacia 17616, and you will find the paper that I wrote in 1994, or published in 1994. That's my PhD thesis. This number, it's actually is called the ATCC. So this is actually ATCC and 17616. And ATCC stands for American Type Culture Collection. Okay, so this is it's a U.S. organization that keeps different bacteria. So you can order bacteria from them, and they collect. And so so that's that's Pseudomonas, and this is what they look like. And when they grow in petri dish, to produce these greenish product, a chemical, and that greenish chemical is helping them to get oxygen. and to bring oxygen to the bacteria. And so that's one way of them to grow faster because it's in a strict aero. They need oxygen and they wouldn't ferment. And so this is what they look like. And in addition, they have what's called a enterodeuterol pathway when they ferment glucose. So what they will do is they take a glucose and they will use a different pathway than glycolysis. So they would go to glucose 6-phosphate, and then they will go to, and then what they will oxidize, and then they will produce 6-phosphogluonic acid. So the end result, though, is this: is 2 ATP. They use 1 ATP, they produce 2 ATP. And the net result is they produce 1 ATP. And if you contrast that to the typical E. coli glycolysis, they take 1 glucose and produce 2 ATP. So energy-wise, Pseudomonas is in a disadvantage because it produces 1 ATP, right? So we think the Pseudomonas probably, this pathway probably is older than glycolysis because it's less efficient. We expect the more efficient, with time, the more efficient, energy efficient method will be found and then take over the less energy efficient. Go ahead with questions, Catherine.
So after the enterodeuterol pathway, you would do like the Krebs cycle?
Yes.
Okay.
So because you still ended up with pyruvate, right? So it's a strict aero, and so it will do Krebs cycle. So that's enterobacteria, Pseudomonas, and then I wanted to tell you a little bit about the purple photobacteria. So in lecture in the fermentation lecture about the microbial energy diversity, we talked about a photosynthesis bacteria, right? Remember, there's a photosynthesis, there's a purple bacteria, and then there's a purple sulfur bacteria. And so purple is where the color is due to the absorbance of light, and sulfur is that they produce sulfur because they use hydrogen sulfide to extract as the donor for electron, and then they produce sulfur, right? And so that's what's left, and but that's what for the electron donation electrons. And the purple non-sulfur bacteria uses other molecules to donate the electron. So this is what they look like, purple sulfur bacteria. So that's really purple, right? So next time when you walk in the Central Park or along the Bronx River or somewhere in the woods, you will see green and then you see purple and say, aha, these purple are not leftover color, it's purple bacteria. And if we were to look, because they're photosynthesis bacteria, remember, the photo centers all sit on the membrane. And so either in stacks of membrane or in vesicles of membrane, or both, you can find both of them. What's the difference between, well, algae is a eukaryote, right? And which we, at the lecture one, we talked about, it's single cell or simpler or in long chains algae, right? So but purple sulfur bacteria is actually bacteria. It's a prokaryote. So those are, that's the main difference. Um, so the purple sulfur bacteria oxidize hydrogen sulfide to element sulfur so that they can extract the electron for NADH, right? So remember the NADH is needed for the Calvin cycle to provide for fixing the CO2, right? From CO2 to glucose, you're missing the hydrogen, right? And the electron. But the purple non-sulfur bacteria are using organic molecule as the donor of electron. And so they could be a whole variety. And so they are not producing sulfur. And that's the difference. Underline the sulfur and then oxidizing bacteria. Yes, photosynthesis, no. Yes, photosynthesis happens in the photocenter. Remember the photoreceptors, they all line up because they are having the electron transport chain, right? And then they pump out the electrons. And so they need to be really in proximity. They need to be next to each other. The only way the bacteria or any living organism to get these things together, either they form a protein complex or they get them in membrane. So instead of floating freely in water, right? And so having all the proteins in a pathway in a membrane, that increases the chance they get together and then they pass on. So electron transport chain is always in the membrane. Okay. The non-sulfur bacteria is they're getting the electron from organic molecule instead of getting it from hydrogen sulfide. So in this case, they are not producing sulfur, right? Because they don't use hydrogen sulfide as a source for electron. Okay, then we have the sulfur oxidizing bacteria. The sulfur oxidizing bacteria, these are the process they go through to extract energy. They can be oxidized. So hydrogen sulfide has lots of energy. They can be used. They can be extracted to donate electrons and from 2 electrons and then 6 electrons, right? And be oxidized. So anything, I know sometimes it's confusing, anything can be oxidized that could be used to generate energy. How would you remember oxidize, reduction? So the thing is, if you take glucose and you can oxidize it, right? Well, how do you know? Well, you can burn it. Anything can be burned. When you burn a paper, what do you generate? You generate completed burning, right? If it's completely burned, you generate CO2, right? And it's the oxidized. Yes, excellent. And so that's the process. So burning generates energy, and burning releases energy from the molecule, right? If you burn wood, the wood has energy. Where's the energy come from? From the wood, in the wood. We know you burn wood and it gives you heat. heat is an energy, right? But where does the energy come from? In the wood. The wood has energy, right? Where does the energy come from? Come on, we just talked about this. Is it from sun? Through photosynthesis, generating NADH, energy, and ATP, and then fix CO2 into glucose, right? Okay, and so that's the what the hopefully help you understand oxidizing, and that's where the energy is generated. And so these are examples of how they look like at individual cell, and you can see them in streams, filamentous sulfur oxidizing bacteria, and these are often found in cold mining area because cold tend to contain hydrogen sulfide. That's an energy source for these sulfur oxidizing bacteria. And along the way, related to sulfur is the sulfate, sulfur reducing bacteria. And what happened is in the anaerobic respiration. So these bacteria does anaerobic respiration. So remember anaerobic respiration? And we remember Krebs cycle, right? That's coupled to aerobic respiration, where aerobic respiration means using oxygen as the final electron receptor, right? What if you don't have oxygen? Some bacteria can use sulfuric acid or sulfur as the electron receptor to producing hydrogen sulfide, right? So these anaerobic respiration, typically they burn glucose. They burn glucose to extract energy. Leftover energy is being, so now you understand the leftover energy is being passed on to hydrogen sulfide, and this hydrogen sulfide then being used as energy source to for sulfur oxidizing bacteria, right? So if you have both of these bacteria living together, would they be happy with each other? They will be very happy, right? So the sulfur oxidizing bacteria will be the most beneficial. They will be saying, oh, okay, can we live together? You can give me hydrogen sulfide and I can burn it for energy. I'll give you this sulfuric acid as your electron receptor in exchange with no energy left. And so we have this cycle going, right? So they can be happy, live happily ever after, because sulfur reducing bacteria will be getting energy from glucose so that it can do anaerobic respiration. The sulfur oxidizing bacteria can use hydrogen sulfide as an energy source, and they will live happily ever after, right? Okay. So here are some sulfur and sulfur oxidizing bacteria. The whole point is that they can be either curved or short rods or in chain. It can be any type. Okay, but they all have these sulfur and sulfate oxidizing ability. Okay, it's not determined by the size or the shape. That's important. It's determined by the enzyme, the presence of the enzyme. And then you can have other bacteria using other ways to extract energy. So there's nitrosulfine bacteria can oxidize ammonia using oxygen, and then you can overall, this is the end product. The nitride is being produced, and then you can actually have the next from here, and then you can have so you can have this being used as the energy source here to further oxidize to uh, uh, nitrate. Okay, so now you have another and a set of energy gets extracted. Now it's less than 275, but still, if you have endless supply, then you can still make a living out of this. And this is how they look like. These bacteria tend to have, so this is nitrobacter, and they tend to have membrane structures because these enzymes are lined up in the membrane. Yes, that's how they express the energy. Ask the chemist why they do that. They okay, and so so then you have another group called methyl-truths, and they use methane, and I think we mentioned that in the past. They use methane they oxidize methane to methanol using an enzyme, methane monooxygenase. And because this bacteria, because you only need this one enzyme to use methane to generate energy, and any bacteria obtains this enzyme, will be able to use that and then be classified as methyl-truths. So then that's why they have a large variety of these bacteria. Now, this is one example of how they look like. They have these enzymes sitting on the membrane, right? So they have lots of membrane structures. We spend a lot of time talking about bacteria. So you learn a lot of bacteria. How many bacteria groups, phyla, we talked about? I'm sorry, we just talked about one bacteria. All right. The next group I wanted to tell you about is phylum 2, the gram-positive bacteria. And the gram-positive bacteria can be slightly simpler. It'd be further regrouped into 5 instead of 17 groups. And some of the things you already know, you see it's high GC or low GC means what? Higher percentage of GC pairs in DNA, right? And then the other criteria they can use to separate gram-positive is whether it's formed spore, non-sporulating or endospore. And then you have this bacteria, which is an exception with no cell wall. Okay, so as a microbiologist, you learned, trying to drill into your head, you never say all, you say mostly, because there's always an exception. If you set a rule, I can always find an exception. So just say, well, most bacteria, almost all bacteria. Okay, do that. That will keep you safe. GC is the GC versus AT. GC content. Is that clear? Okay, yeah, so it's a low GC, high GC. E. coli is 50-50, right? 50% GCs and 50% ATs. Okay, so the non-spoilating low GC is these 3 groups. Staphylococcus, that's on your skin. Streptococcus, that's in your throat. Lactobacillus, that's in your small intestinal tract. Okay, it's all over. this is what they look like. Staphylococcus. Okay, it forms like a grape, a cluster of grape. Actually, in Chinese, this was they actually used the grape like bacteria, and the streptococcus they translated into a chain-like bacteria, and it's more literal, right? And so this is how they divide. The main difference is that streptococcus, when they divide, you can see they divide in the same plane, okay? You see, it's parallel. They always divide this way. But if you look at the staphylococcus, and they can be dividing randomly. They divide this way or that way. And the net result is it's random and it forms a cluster. They don't form a chain. And this is lactobacillus, and this is the bacteria that's in your yogurt. And when you eat, and that's what you have, and this is what's probiotic, and this is you resupplying your small intestinal tract with this beneficial bacteria. All right. Then the next group is endospore forming bacteria called the Bacillus thuringiensis. That's what we want to talk about. So Bacillus is a group that forms the endospore. And then among this family, there's one particular bacteria worth noting, which we'll come back to talk about, is the this bacteria forms a perispiral crystal. So it has this endospore, but it also produces this crystal. Okay, and this crystal is actually toxic to many of the insect larvae. And traditionally, peasants have they grow this bacteria on purpose, and then these bacteria they dry these bacteria and now become powder, and then they sprinkle this powder on corn. And so when insect larvae eats these crystal, they will get killed. And this is, just keep a note, in the next lecture we'll talk about that. Why is that important? And so that's Bacillus. And then there's another bacteria that you might have heard about in part. It's called the Clostridium botulinum. This bacteria has no cytochrome system, so it cannot doesn't have the electron transport chain, so it can only get energy from fermentation, right? So it does fermentation only, and it's related to botulism, and it produces a toxin that relaxes muscle. We'll talk about that in the future, but for now, it relaxes. It's relaxed the muscle, and it prevents the muscle from contract. Have you heard about this bacteria in some form? Have you heard about Botox? Yeah, well, Botox is this. It's produced by a bacteria, and it relaxes the muscle. The theory is your muscle you form wrinkle because your muscle is tight, and you inject inject your toxin, and the muscle relax, and your wrinkle goes away. Um, that's what they use for. But obviously, if you inject too much, and if this bacteria gets in your gut and produce so much toxin and circulate in your blood, and your heart relax and doesn't retract, what happened? You die, right? Your heart needs to pump. Okay, so the next, I think I wanted to show you some example of what a bacteria called wall-less bacteria and mycoplasma. And this is a unique bacteria. It has no cell wall. It doesn't produce cell wall. So if this bacteria doesn't produce cell wall, can you use penicillin to kill this bacteria? No. Thank you, Catherine. No. Yes. Um, and uh, and this is what they look like. So is this one bacteria or many different types of bacteria? If I didn't show you the topic, and you would say, oh, this looks like a different bacteria, right? Because they're different size and shapes. And could be a staphylococcus. This could be some long chain. This could be something, right? Right? And this could be streptococcus, and right. And so because it doesn't have cell wall and it's just randomly, it's a lot random, the cell shape. Do they have? No, they don't have cell wall. That means they have cell membrane. The cell wall, the function of cell wall really is to provide a rigid structure. It does have, so this bacteria, if you put this bacteria in soil, they may die. Well, because the salt concentration changes dramatically, right? And so they live in environment very stable. So they don't have to worry about the cell rigidity, how strong. And when they grow, because it doesn't have cell wall, they form a more or less flat pile, instead of, like if you use brick, they tend to form, they form a pile. But if you use balloon, water balloon, they tend to be flat, right? That's what the difference is. And you can also have filamentous bacteria, streptomyces. And streptomyces, a lot like fungi, it has high GC. So this is what they look like. And but this is a more important, because streptomyces are typed or differentiated based on their reproductive structure, the spore bearing. They are exospores versus endo. Okay, so these are exospore, and they have different structures, how they produce exospore. And the steps of producing exospore starts from, then they have different forms. And so they have a growth, and then the tip will curl, and then they self-form partitions, and then they self-form walls, and then these are then matured. Once these matured, these have special names. They call conidia sport, and these spores can, once they mature, the wing can just blow, okay, and then they will spread. Okay, now this is what they look like. So compared to fungi, have these long strings, right? Like your bread mold, right? Mold has these long stringy cotton-like structures, right? And the streptomyces, they have these very short. They usually on the surface. So these these hyphas, they're very short, okay, just on the surface. And so then these uh, these black areas, the reason they're black is because they're producing endo or exospore. So it is extremely important that when you see these, don't open them. Certainly don't blow them, because if you blow, the spore will be everywhere, and it's really difficult to get rid of. Okay. So the last high GC bacteria I wanted to talk a little bit is called the Mycobacteria tuberculosis. You heard about tuberculosis, right? It's a disease, and it's a bacteria that causes this disease. What's unique about this bacteria, you might have, I think we covered it in that it's called fast acid staining. Anyone remember? Fast acid staining. Use acid to break down and to stain the bacteria. This is the only bacteria will stain positive stain for acid fast staining is this mycobacteria, because every other bacteria simply gets degraded in acidic condition. Except this bacteria has this malic acid layer. It's like a wax. This is the I'm going to use the face mask N95. In the past, when I've lectured this, even 2 years ago, if I say face mask N95, people have very difficult imagine. But now you know face mask N95, right? The difference is N95 is great. It filters out everything. It protects you from all things, virus, COVID-19. But it's hugely difficult to breathe through. It's virtually impossible to lecture through an N95 face mask. So it will be very challenging. I'll be out of the breath. Okay, and so this malic acid layer is hugely protective for the bacteria. So any our immune cells or enzyme, they cannot get through that layer. So it protects the bacteria from this attack or our immune system. So we cannot kill them. This is a pathogen, is a bacteria pathogen. Okay, and when you have it, it will cause disease. If you breathe into your lung, it's really difficult to get rid of. And you'll be on antibiotic for 6 months at a time. Because it has this malic acid layer, we now have some drugs that can reduce the production of this protective layer. Now, having this protective layer is good for the bacteria so they cannot be killed, but the side effect is that they cannot get enough nutrient in as well. So they grow very, 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 very slow. How slow? So for a bacteria to form a colony, that visible pile, E. coli takes one overnight, like 16 hours, 18 hours, you get a colony. For this bacteria, you need 2-3 weeks to get a colony. Imagine you put them on, and you have to wait 2-3 weeks before you actually see a colony. All right. And so that they grow very slow and make it very difficult to study and very difficult to kill. Later on, we'll talk about the actual disease, what they cause, and but for now, remember this part. Now, obviously, there are lots of other bacteria I didn't get to talk about, so I know I wouldn't be able to talk about, so that's why I put them in as your homework. So this is your homework of looking up the energy source, the carbon source, the electron source, the characteristics of these bacteria. Okay. So back to the question, I can't read the chat, but yes. So mycobacteria, the mycolic acid has the protection layer. It offers the bacteria protection, so from human immune system, but it also prevents them from taking up nutrient efficiently, so they grow very slowly. They have a very slow growth process. Okay, but in the long run, if we think about this bacteria, it's more important for them to survive, and in the long, that's where they grow. And then they could persist for decades, right? So up to this point, when we talked about the microbial diversity, we talked about bacteria. We said there were 17 phyla, and we talked extensively about phylum 1, the proteobacteria. We talked a little bit more about gram-positive bacteria. I left homework for some of the rest of the bacteria for you to look up. And I will talk a little bit about the archaea that we know about. So archaea can be further divided into 3 phyla. And what's common about them is it has no peptidoglycan. It has pseudopeptidoglycan. And the difference is that the repeating units doesn't form a smooth structure. So the cell wall shape and size could be random, even within the same species bacteria. And their membrane is different. It's linked lipid, and the RNA polymerase is complex. So there are 3 different phyla, and you have Eukaryota, and it includes methanogenes, halophiles, and then you have Archaeota. That's a phyla that lives in high temperature or low temperature. And then you have this new proposed phyla that it's proposed because this bacteria can only be detected when we sequence environmental DNA. We know they're there, but we haven't figured out how to grow them. All right. So that's the third on the phylum. So here are some examples of methanogenes, and you see the shape is even within the same species. It looks different, not like E. coli have very reproducing structure. And so here's the SCM picture of how you cross-section on TEM picture. And here are examples of halophile. Now, they tend to live in high salt. Halo means high salt, and this is when they dry seawater to make salt. And so as they as the salt concentration increase, the color actually changes to the red, and you can see bacteria and the crystal, salt crystal. And the reason they're red is because these bacteria rhodopsin. So these halophiles, remember when we talk about the photosynthesis, they're an example of this one single protein, bacterial rhodopsin, that's being that function as the photosynthesis, and it takes out, gets the energy from light, and then pumps out electron, and to form electron gradient. I'm sorry, proton gradient. So the proton is outside, and then it comes in. And so as long as it's not a very efficient process, right? It's a single protein and pumps out one proton at a time, but sunlight is limitless, and so they just sit there and keep producing the ATP. That's how they can. That's how they grow. All right, and this is an SEM picture. I'm sorry, keep saying this is a TEM picture. It's a section, and then you have some of the membranes, vesicles. So this one is the shape of is random, even even for this one bacteria cell. All right, and so so that's what they look like, and so this is really the first lecture of this series, focusing on surveying the microbial environment with different microbes, and so I hope to that this discussion has opened your view of the microbial world a little bit more, and there are more than E. coli, and your closest relative to E. coli is what's the closest relative or microbe? Yeah, Salmonella is the one that very close to E. coli, and but then there are distance, and then you have these archaea could be live in extreme environment. Okay, so I'm gonna end the recording.